Heredity is next.
Meiosis ensures the formation of haploid gamete cells, which have one set of chromosomes, in sexually reproducing diploid organisms. Diploid means two full sets of chromosomes, where the cell contains one from each parent. It results in daughter cells with half of the number of chromosomes as the parent cell, as diploid parent produces 4 haploid sex cells. Meiosis involves 2 rounds. The first round is meiosis 1. Phase 1 is where the nuclear envelope disappears, the fibers begin to form, and the DNA coils into visible chromosomes. The difference with mitosis is that these chromosomes are paired. The chromatids exchange genetic information with chromatids from their other chromosomes. This produces recombinant chromatids. Metaphase 1 is where the fibers align across the center of the cell, but the double chromosomes remain in pairs. Anaphase 1 is where the fibers separate the chromosomes. And in telophase 1, the nuclear envelope appears again. Each nucleus contains only one double chromosome from each pair. Lastly, cytokinesis separates the cell. The daughter cells are haploid and are genetically different from the other, and also different from the parent cell. Meiosis 2 is the same, except now the metaphase, you can see that the double chromosomes are now aligned. Then in anaphase, the fibers separate the sister chromatids, and anaphase 2 and telophase 2 and cytokinesis happens. The daughter cells are haploid here as well. Variation is caused by the law of segregation, which is a separation of alleles during gamete formation. Each gamete receives only one parental allele. This allows for more varied combos of alleles when fertilization. It is also caused by independent assortment, where genes for 2 or more traits are sorted into gametes independently and the genes are not linked. Inheritance is random. Also, any genetically unique sperm can join with any genetically unique egg, which means that fertilization is completely random. Non-disjunction is the failure of chromosomes to fully separate during the formation of gametes. Results in too many or too few chromosomes. Heritable information provides for the continuity of life. Nucleic acids are carriers of genetic info and is transferred to the other cells during cell division. All organisms use nucleic acids and ribosomes to synthesize proteins based on those nucleic acids and use metabolic pathways. This means that if you add a gene that codes for something in this random cell, the cell can produce it, since all cells have ribosomes. Genes determine traits. Alleles are specific variations of genes represented by capital and lowercase letters. Phenotypes can be homozygous or heterozygous. Phenotypes are the physical result of the genotype. For probability, if it says and, multiply. If it says or and they are both mutually exclusive, then it cannot happen at the same time, then add them. Monohybrid and dihybrid crosses can be modeled with Punnett squares showing possible offspring. Patterns of inheritance can be modeled with pedigree. Autosomal dominant traits show affected offspring with affected parents. Autosomal recessive genes show affected offspring with unaffected parents. Hypothesis testing helps determine if differences in numeric data are due to independent variable or just by chance. Null states that there is no relationship, and alternative states that it's due to a non-random cause. Plug everything into the chi-squared goodness of fit test and find out the degrees of freedom by taking the number of distinct possible outcomes minus 1. Y-squared is larger than the critical value. That means it's not due to chance, and you reject the null hypothesis. In reality, many traits do not follow the ratios predicted by Mendel's laws, as some genes are adjacent and close to one another on the same chromosome, and they are called linked genes. They are less likely to be separated during crossing over. Traits that are determined by genes located on sex chromosomes are sex-linked traits. Map distance tells you how close together a pair of linked genes are. Linked genes usually have a recombination frequency of less than 50%. A pair of linked genes have a recombination frequency of 30%. That means that they are 30 map units apart. Some traits are also caused from non-nuclear inheritance and from mitochondrial or chloroplasts. These traits are maternally linked. Some traits are the product of multiple genes, like hair color. Back to the pedigree, sex-linked recessive shows affected offspring with unaffected parents and males being more affected than females. Maternal inheritance shows every offspring from an affected female being affected. The same genotype can result from multiple phenotypes, as environmental factors can influence gene expression. It shows the concept of phenotypic plasticity, the ability of one genotype to produce more than one phenotype.